The biological continuum of human reproduction, from the microscopic fusion of cells to the delivery of a neonate, represents one of the most perilous physiological gauntlets in mammalian biology. It is a highly fortified obstacle course where the margin for error is razor thin. That obstacle course begins immediately upon ejaculation. Millions of highly motile sperm are deposited into the vaginal vault, an environment so acidic it acts as a lethal immunological defense against foreign pathogens. To survive, the sperm rely on surging maternal estrogen levels, which briefly alter the cervical mucus, turning it from an impenetrable barrier into a slippery, nutrient-rich reservoir that allows the most robust cells to swim upward. But reaching the fallopian tubes isn't enough. The sperm must undergo capacitation, a mandatory maturation phase triggered by the female reproductive tract. Here, the sperm literally shed stabilizing proteins and cholesterol from their outer membranes, transforming into hyperactivated, highly fluid biological drills. When these hyperactivated sperm finally reach the egg, two distinct mechanisms execute in rapid succession. First, the acrosome reaction deploys potent enzymes to drill directly through the egg's thick outer shell. The exact microsecond a single sperm breaches the membrane, it triggers the cortical reaction. The egg fires an immediate electrical and chemical seal across its surface, instantly rendering it impermeable to any other sperm to prevent lethal chromosomal abnormalities. This successful fusion restores the cell's diploid chromosome number. Over the next 7 days, it rapidly divides from a single cell into a complex, fluid-filled structure called a blastocyst, which sweeps down into the uterus and aggressively burrows into the prepared endometrial lining. With implantation complete, the blastocyst begins the construction of the placenta. This invasive cluster of cells now serves as the master regulator of the pregnancy, beginning an irreversible clock that will soon require the maternal body to undergo a total physiological reorganization. The growing embryo operates in a hypermetabolic state. To sustain its intense developmental demands, it chemically hijacks the mother's baseline physiology, forcing virtually every maternal organ system to increase its functional capacity. This side-by-side data visualization illustrates the scale of this maternal stress. To ensure enough low-resistance blood flow reaches the placenta, the mother's blood plasma volume expands by 50%, nearly 1,600 milliliters of extra fluid. To pump that heavy fluid load, her resting cardiac output spikes, driving her heart rate into a continuous sinus tachycardia. While the mother's cardiovascular system stretches to its limits, the fetus spends the first trimester in an explosive phase of organogenesis. By week 6, the neural tube closes to form the precursor to the brain and spinal cord, and a primitive heart tube begins beating at 110 beats per minute. Moving into the second and third trimesters, the biological focus shifts from building structures to maturing them. The soft cartilaginous skeleton undergoes rigid ossification, the lungs practice rhythmic breathing movements, and the fetus rapidly packs on dense layers of subcutaneous fat required for thermal insulation outside the womb. After 40 weeks, the fetus is biologically equipped for independent life, but its sheer size and mass, averaging 7.5 pounds, now trigger an impossible biomechanical crisis for the mother. To initiate the exit sequence, the maternal immune system floods the uterine tissues with a cytokine storm, a massive localized inflammatory reaction. These aggressive chemicals physically degrade the amniotic sac until it ruptures and break down the collagen in the rigid cervix, forcing it to soften and dilate. The mechanical success of labor depends on the 3 Ps: power, passenger, and passage. This geometric diagram illustrates the core physical problem. The wide transverse diameter of the fetal skull and shoulders is naturally incongruent with the narrow, rigid, and fixed dimensions of the maternal pelvic ring. The infant cannot simply be pushed straight out. Instead, the fetus must execute a mandatory, precise sequence of 3-dimensional rotations. Upon hitting the resistance of the pelvic floor, the infant performs the first cardinal movement, sharp cervical flexion. By tucking its chin tightly against its chest, it presents the absolute smallest possible diameter of its skull to the birth canal. Driven deeper by intense uterine contractions, the fetus must then rotate internally 90 degrees, turning its face toward the maternal spine. This exact rotation aligns the broad span of its shoulders with the widest part of the maternal pelvis, allowing the neck to extend upward and slip beneath the pubic bone for delivery. Successful delivery is a specific, mathematically driven sequence. When the mechanics of the passenger and passage fall out of alignment by even a single millimeter, the process stalls, immediately endangering the fetal oxygen supply and the integrity of the maternal tissues. The exact moment of birth forces the neonate through an acute physiological shock. The infant must instantly abandon its fluid-filled, placenta-dependent environment and aggressively expand its own lungs to pull in atmospheric air. To secure this cardiopulmonary transition, clinical guidelines require delaying the clamping of the umbilical cord for at least 60 seconds. This calculated pause allows the placenta to continue transfusing critical blood volume into the infant, easing the massive pressure required to fully open the neonatal pulmonary vascular bed. Simultaneously, the newborn faces extreme internal vulnerabilities. Because neonates lack a mature intestinal microbiome, they are born entirely deficient in vitamin K, an absolute biological requirement for synthesizing blood coagulation factors. A standard intramuscular injection of vitamin K is universally administered within hours to prevent catastrophic, undetectable intracranial bleeding. These rigorous, immediate clinical interventions successfully stabilize the infant's acute transition. But once the cardiopulmonary system is secure, the battle for survival shifts entirely to a new external environmental frontier. The first few years of human life are categorized by explosive, rapid neurological wiring. This delicate brain architecture is built largely in response to direct caregiver interaction, proper nutrition, and strict physical safety. During periods of sleep, however, the infant's arousal and respiratory systems remain highly fragile. This minimalist image reflects the uncompromising, evidence-based standard to prevent sudden infant death syndrome. An infant must be placed exclusively in a supine position, on its back, upon a firm, flat, entirely bare surface. Pediatric guidelines explicitly prohibit weighted garments, loose blankets, or shared adult sleep surfaces. These items physically restrict natural thoracic expansion and alter the infant's arousal threshold, turning a normal resting state into lethal hypoxia. The journey from conception to toddlerhood is a relentless evolutionary gauntlet. It requires massive systemic sacrifice from the mother and exact biomechanical precision from the infant. And when human biology falters under the weight of these demands, meticulous clinical science steps in to safeguard the ultimate triumph of independent life.